Gene family, a group of genes that code for related or identical polypeptides. Share a common ancestor. A gene family originates when a gene is duplicated. Gene paralogs are genes with similar sequences from within the same species. Gene paralogs are genes with similar sequences from within the same species. Gene orthologs are genes with similar sequences in different species. For example, globin gene family. First, let's learn about classification of gene families. Multi-gene families consist of members with similar sequences and functions, though a high degree of divergence does not lead to the removal of a gene from a gene family. Divided into two: simple multi-gene family, complex multi-gene family. Developmentally controlled complex multi-gene family. Simple multi-gene family, complex multi-gene family, and a developmentally controlled complex multi-gene family. Then superfamilies, much larger than single multi-gene families. Much larger than single multi-gene families. Contain up to hundreds of genes, including multiple multi-gene families as well as single individual gene members. Simple multi-gene family, rRNA gene in Xenopus. Simple multi-gene family, for example, rRNA gene in Xenopus. Simple multi-gene family is one in which one or few genes are repeated in a tandem array. One or few genes are repeated in a tandem array. Set of genes for 5SR RNA in Xenopus levis is an example. Set of genes for 5SR RNA in Xenopus levis is an example. Each 5SR RNA sequence is separated from adjacent sequence by a spacer to form a gene cluster. Each 5SR RNA gene is transcribed as a separate RNA molecule that is processed to generate the finished 5SR RNA molecule. Each 5S RNA sequence is separated from adjacent sequence by a spacer to form a gene cluster. Each 5SR RNA gene is transcribed as a separate RNA molecule that is processed to generate the finished 5SR RNA molecule. 5.8S, 18.S, and 28SR RNA genes of Xenopus comprises another simple multi-gene family. Then complex multi-gene family. Organization of histone genes in sea urchin. Histone genes that encode non-polyadenylated mRNA include two families. Histone genes that encode non-polyadenylated mRNA include two families. So we are learning about complex multi-gene family. Organization of histone genes in sea urchin. Histone genes that encode non-polyadenylated mRNAs include two families: alpha histone genes expressed in unfertilized egg and expressed throughout early embryogenesis and silenced by the gastrula stage. Then late histone gene expressed at a very low rate during early embryogenesis, usually expressed after gastrula stage. Then tRNA gene clusters of Drosophila. tRNA gene clusters of Drosophila. The tRNA gene clusters of Drosophila has two characteristics. There are several copies of the same gene in the cluster. There are several copies. The tRNA gene cluster of Drosophila has two characteristics. There are several copies of the same gene in the cluster, and not every tRNA gene is in the cluster. Each of the gene in this cluster is separately transcribed and both directions of transcriptions are used. Then globin gene family. Globin genes are descended by duplication and mutation from an ancestral gene that had 3 exons. Globin genes are descended by duplication and mutation from an ancestral gene that had 3 exons. Ancestral gene gave rise to like hemoglobin, myoglobin, alpha and beta globin genes. Heme binding domain is there. And we try to learn about the structure. An exon structure of globin genes corresponds with protein function, but like hemoglobin has an extra intron in the central domain. the exon structure of globin genes corresponding with protein function, but like hemoglobin has an extra intron in the central domain. When we try to learn about the structure, we learned earlier that all this process of mutation is happening from an ancestral gene that had 3 exons. Remember that point, 3 exons, and from that ancestral gene which have 3 exons means coding sequences. Those exon portions, another 3 ancestral gene produced these 3 types: leg hemoglobin, myoglobin, and alpha and beta globin genes. Okay, so when we try to learn about this picture, 3 exon regions: exon, intron, exon. Along with that, a heme binding domain will be there. This is the globin. Then extra intron leg hemoglobin domain is divided. The exon structure of globin genes. The exon structure, we know where is that exon structure, represented as E in this figure. The exon structure of globin genes corresponds with protein function, but leg hemoglobin has an extra intron in the central domain. Here we could see a picture which shows this: heme, beta globin, hemoglobin, beta globin, and alpha globin portions. Alpha globin gene family, chromosome 16. Beta globin gene family, chromosome 11. Then embryo, fetus, and adult. Then embryo, fetus, and adult. The human alpha globin and beta globin gene families. And evolution of globin gene. We are going to learn about evolution of globin genes. Chromosome 22 MB, chromosome 16, chromosome 11, myoglobin, alpha globins and beta globins, hemoglobins, ancestral globin, non-functional pseudogenes. Ancestral globin turned to myoglobin and hemoglobin. This hemoglobin is of 2 types, which are the alpha globins and beta globins. Exon fusion, duplication and divergence. Exon fusion, duplication and divergence. Separation of genes, expansion of clusters, separate clusters for mammals and birds. So linked alpha and beta genes, amphibians, fish. Single globin gene, lamprey and hagfish, ancestral globin, myoglobin, leg hemoglobin regions. So exon fusion, leg hemoglobin, and along with that duplication and divergence. Then single globin gene, lamprey and hagfish, ancestral globin, myoglobin. Then linked alpha and beta genes, amphibians or fish. Then separation of genes, expansion of clusters. Which are the processes? Exon fusion, duplication and divergence. Exon fusion, duplication and divergence, separation of genes. Then expansion of clusters. Exon fusion, duplication and divergence, separation of genes, expansion of clusters. Next is about pseudogenes. Copies of functional genes that have altered or missing regions such that they presumably do not produce functional protein or RNA products. Many pseudogenes have frame shift or nonsense mutations that disable their protein coding functionality. So these are copies of functional genes that have altered or missing regions such that they presumably do not produce functional protein or RNA products. They are not capable of producing functional protein or RNA products. Many pseudogenes have frame shift or nonsense mutations that disable their protein coding functionality. Then some categories are also there. Processed pseudogenes. Processed pseudogenes result from the reverse transcription of mature mRNA transcripts into cDNA copies, followed by their integration into the genome. Processed pseudogenes result from the reverse transcription of mature mRNA transcripts into cDNA copies, followed by their integration into the genome. Ribosomal protein pseudogenes. Then non-processed pseudogenes arise from inactivating mutations in one copy of a multiple copy or single copy gene, or from incomplete duplication of an active gene. So processed pseudogenes result from the reverse transcription of mature mRNA transcripts into cDNA copies, followed by their integration into the genome. Ribosomal protein pseudogenes, they undergo reverse transcription of mature mRNA transcripts into cDNA copies, and later integration of that particular part into genome will be taking place. That is the example for processed pseudogenes, ribosomal protein pseudogenes, processed type of pseudogenes. The non-processed pseudogenes arise from inactivating mutations in one copy of a multiple copy or single copy gene, arise from inactivating mutations in one copy of a multiple copy or single copy gene, or from incomplete duplication of an active gene. Then evolutionary clocks. These are the genetic sequences within genes that can help determine when the past species diverged from a common ancestor. Discovered in 1962 by Linnaeus Pauling and Emily Zuckerkund. And the number of differences in the nucleotide sequences of the hemoglobin protein signifies a certain amount of time. The number of differences in the nucleotide sequence of the hemoglobin protein signifies a certain amount of time that has passed since the two species split from the common ancestor. So, duplication, rearrangement, and mutation of DNA contribute to genome evolution. The basis of change at the genomic level is mutation, which underlies much of genome evolution. The earliest form of life likely had a minimal number of genes, including only those necessary for survival and reproduction. The basis of change at the genomic level is mutation, which underlies much of genome evolution. Then the size of genomes has increased over evolutionary time. The size of genomes has increased over time, evolutionary time, with the extra genetic material providing raw material for gene diversification. So, the earliest forms of life likely had a minimal number of genes. Later, that just increased evolutionarily, time related, and that extra genetic material is acting as a raw material for genetic diversification. Okay. So, here we could see the basic structure of a gene with control flanking region along with exon 1, later in intron 1, exon 2, intron 2, exon 3 flanking region. So this exon 1 portion has a transcription initiation. So basic structure of a gene consists of control flanking regions along with some exons, exon 1, exon 2 and exon 3 along with some introns, and intron 1 and intron 2. So this intron portions are also there. So exon portion contain 3 regions initially. Like here we could see a transcription initiation portion. Then initiation codon will be there. The GT region. Same is the case with the exon 3, AG stop codon portions. 3 portions are there. Then poly A addition site, flanking region. Which are the major portions? Exons and introns. Okay. Along with some control flanking regions. Then transcription initiation, stop codon, poly A addition site, etc. can be seen. Common mechanisms of evolution of gene families. We are learning about evolution of gene families. So there must be some mechanisms which help in the evolution of gene families, like rearrangement of parts of genes, exon duplication, errors in meiosis, exon shuffling. Exon duplication, errors in meiosis, and exon shuffling. These are the 3 mechanisms by which gene families undergo evolution. So first is about this 3 points of exon duplication, errors in meiosis, and exon shuffling comes under the portion of rearrangements of parts of genes. Later, duplication of entire genome. Gene. Duplication of entire gene or chromosome sets and divergence, that is polyploidy. Then unequal crossing over and gene conversion. Then transposable elements. Once again, rearrangements of parts of genes, duplication of entire gene, unequal crossing over and gene conversion, transposable elements. Then genome size increases through duplication of exons, genes, gene families, and entire genomes. Genome size increases through duplication of exons, genes, gene families, and entire genome. At lowest level, exons duplicate or shuffle. Gene A is there, introns are there, exon duplication, new gene variant is formed. Then exon shuffling will be taking place. With the help of that also some new genes are created. Okay, gene A undergoes some exon duplication to create a new gene variant. Gene B undergoes some exon shuffling. Gene A undergoes some exon shuffling to create a new gene. Okay. Then at next level, entire genes duplicate and create multi-gene families. Gene A undergoes gene duplication to create a multi-gene family of this gene A. So at next level, gene families duplicate to produce gene superfamilies. So here we could see gene A, gene B, gene C are arranged. Then gene A, gene B1, gene C1. Duplication of gene families will be taking place. And gene A2, gene B2, gene C2. Chromosome 2. The entire genome duplicates, doubling the number of copies of every gene and gene family. The entire genome duplicates. Okay. Then next, the entire genome duplicates, doubling the number of copies of every gene and the gene family. Entire genome duplicates. So when we are going to learn this process, first we need to know about the exon duplication and exon shuffling. Later, new gene variant is formed, new gene is formed, then multi-gene families are created because of amplification or to a greater extent duplication, gene duplication. Then superfamilies, gene superfamilies are produced. After that, entire genome is going to duplicate. So genome size increases through duplication of exons, genes, gene families, and entire genomes. Rearrangements of parts of gene exon duplication, errors in meiosis, and exon shuffling. The duplication or repositioning of exons has contributed to genome evolution. The duplication or repositioning of exons. First, the duplication or repositioning of exons has contributed to genome evolution. Errors in meiosis can result in an exon being duplicated on one chromosome and deleted from the homologous chromosome. Meiosis. Errors in meiosis. Then in exon shuffling, errors in meiotic recombination lead to some mixing and matching of exons either within a gene or between two non-allelic genes. Within a gene or between two non-allelic genes. Okay. So exon shuffling may give rise to new genes. Tissue plasminogen activator. Exon shuffling may give rise to new genes. Tissue plasminogen activator TPA. Tissue plasminogen activator, finger module, growth factor module, Kringle module, plasminogen is there, fibronectin, epidermal growth factor, and portions of ancestral gene is there. Epidermal growth factor gene with multiple EGF exons. Exon shuffling will be taking place. Then fibronectin gene with multiple finger exons. Exon shuffling. Then gene duplication. So plasminogen gene with the Kringle exon. Then later exon shuffling will be taking place. And some exon duplication is also accompany this process. So TPA, tissue plasminogen activator gene has informed the TPA, tissue plasminogen activator gene has formed by exon shuffling and duplication of three ancestral genes. Tissue plasminogen activator gene has formed by exon shuffling and duplication of three ancestral genes. Then duplication of entire gene can create multi-gene families. Tandem gene family. Duplication of entire gene can create multi-gene families. Tandem gene family. Members of the multi-gene family are clustered on the same chromosome. Then dispersed gene family. Members of the multi-gene family are on different chromosomes. Chromosome 1, chromosome 2, chromosome 3 and chromosome 4. Tandem gene family and dispersed gene family. These are the two types of gene families. Tandem gene family in the sense Members of the multigene family are clustered on the same chromosome, but dispersed gene family. Members of the multigene family are on different chromosomes. Tandem gene family and dispersed gene family. These are the two types of duplications of entire gene can create multigene families. Duplication process, not amplification. Just some processes of duplication. This create superfamilies, multigene families with two kinds of systems: tandem gene family and dispersed gene family. Then duplication of one or entire chromosome sets. Duplication of one or entire chromosome sets. Accidents in meiosis can lead to one or more extra sets of chromosomes, a condition known as polyploidy or aneuploidy. Accidents in meiosis can lead to one or more extra sets of chromosomes, a condition known as polyploidy or aneuploidy. The genes in one or more of the extra sets can diverge by accumulating mutations. These variations may persist even if the organism carrying them survives and reproduces. An unequal crossing over can expand and contract gene numbers in multigene families. Unequal crossing over can expand and contract gene numbers in multigene families. Unequal crossing over can expand. Unequal crossing over. Unequal crossing over can expand and contract gene numbers in multigene families. Number of members in this cluster increase, number of members in this cluster decrease. Unequal crossing over will taking place. This is happening because of problems in mutation. Meiosis, mutation in meiosis. Just sorry, difficulties or problems in meiosis. Unequal crossing over gene family ABC, ABC, ABC. During synapses, gene A aligns with gene C because of homology between these two. Then a portion of gene C is changed to the sequence of gene A. Gene A remains unchanged. Only gene C has a portion dark of the gene A sequence, converted region. Alternative resolution, unequal crossing over. Then divergence. Intergenic gene conversion can increase variation among members of a multigene family. Intergenic gene conversion can increase variation among members of a multigene family. One gene is changed, the other is not. Then gene conversion. Concerted evolution, jointed evolution can lead to gene homogeneity. Gene conversion will be taking place and crossover is happening. So how transposable elements contribute to gene family or genome evolution? Multiple copies of similar transposable elements may facilitate recombination or crossing over between different chromosomes. Insertion of transposable elements within a protein coding sequence may block protein production. Insertion of transposable elements within a regulatory sequence may increase or decrease protein production. Insertion of transposable element within a regulatory sequence may increase or decrease protein production. Then transposable elements may carry a gene or groups of genes to a new position. Then transposable elements may carry a gene or groups of genes to a new position. Transposable elements may also create Transposable elements may carry a gene or groups of genes to a new position. Transposable elements may also create new sites for alternative splicing in an RNA transcript. In all cases, changes are usually detrimental but may occasionally prove advantageous to an organism. Then that's all about this topic. Later, we'll be learning about some other aspects of this topic. Okay, thank you.